Hi everybody, it's Mrs. Mancuso, and we are moving on to Unit 5 in AP Bio. So I'm going to go through this big idea sheet and make sure that you write down any questions that you have so that you can ask me during our Zoom on Thursday. All right, what are the main differences between mitosis and meiosis? All right, so mitosis is just for asexual reproduction or for growth or repair. It gives you identical cells. Whereas meiosis is how you make gametes. And gametes are for sexual reproduction. So we're talking about two parents coming together to make the zygote. And the only way you can make gametes is to go through the process of meiosis. All right, how does meiosis produce haploid gametes? All right, first of all, it's going to go through 2 rounds of division. So you still go through S, which is the replication of DNA, but then you actually divide your chromosomes twice. So what happens in meiosis 1 is that your chromosomes pair up. So the pairing up is going to come up in a minute. I know I have it as a later question. So in round 1, these pair up and the pairs separate. So again, you in your cells are all diploid. That means you have 2 of each type of chromosome. One of them came originally from your mom's egg, and one came originally from the dad's sperm. So those 2 together is what gave you your pair of chromosomes. And then in meiosis 2, the sister chromatids separate. And if you think about it, in mitosis, we only separated sister chromatids. We lined them all up and we separated them. But here in meiosis, you have 2 rounds so that you are lining up the pairs, separating the pairs, and then lining up the sister chromatids and separating the sister chromatids. All right, what's the significance for a species to produce haploid gametes? So the only way that you can have sexual reproduction, so 2 organisms, is that if they are each providing a half. So a half and a half is a haploid and a haploid, and together they make a diploid zygote. So that's the only way you can get a complement from 2 different organisms, is if they each provide half of the genetic information, half the chromosomes, half the DNA. All right, what is synapsis? So this is the word that I said was going to come up again. So this is the pairing of homologous chromosomes. It happens during prophase 1. It looks like this. So this could be chromosome 1 from mom and chromosome 1 from dad. And they pair up here at the equator so that they will separate. And together, you call it a tetrad because there's 4 sister chromatids. And why is it important? Because you want each gamete to receive part of the homologous pair from each parent. To receive 1 chromosome from each parent. It's also pretty significant and important because you only pass on half of your genes to your offspring. The other half is going to come from the other parent. So if you think big picture why this is important, the reason is variety. Variety in the offspring is due to sexual reproduction. So if there wasn't this unique combination that came together to make each offspring, and if they were identical, then that would be asexual reproduction. But instead, there's a lot of variety in the offspring, and we know that that increases the chance of survival in your species as a whole if the members of the species are each a little bit unique. So no matter what the population has to go through, there might be some individual that might be able to survive it based on their genes. All right, what is crossing over and why is it significant? So first of all, crossing over is when you exchange DNA between homologous chromosomes. And so let me draw a picture. Here's one chromosome. Here's my homologous pair. And during synapsis, they get super close. And right here, if these exchanged, what it would look like is this. And so you see that new combination happening. So when they separate, one of the gametes is going to look like this, and the other one is going to look like that. This is the only way to separate linked genes. So I always use that example of linked genes like freckles and red hair. And so most oftenly you see those traits inherited together because they're on the same chromosome. The only way that you would separate it and not see them together is if crossing over happened. So again, this also increases variety. That's the significance of it. Gives you a new combination of genes. New combo of genes. Okay, what is Mendel's law of segregation and independent assortment? So segregation is that your chromosomes, your homologous chromosomes, line up and separate so that you are making haploid cells. So for example, if this is the parent, it's why when you do a Punnett square, you do this and this. The alleles will separate. And half are going to get the big A and half are going to get the little a of all the gametes that you make. And independent assortment is they line up independent of each other. And what I mean by that is the way that they line up does not depend on if they're a maternal chromosome or if they're paternal or if they're number 2 or number 17. They all just line up with their pair and then separate. And so what that does in terms of significance is greatly increase variety because there's so many different combos that you could get. A lot of combinations. Okay, these two laws are limited to what type of genes? Okay, these are limited to genes that are on different chromosomes. If two genes are on the same chromosome, you do not separate them. So the law of segregation does not apply to them, and they're not going to line up separately because they're on the same chromosome. So these only apply to genes that are found on different chromosomes. All right, what's the significance of fertilization? Okay, fertilization restores the diploid number. So you know as humans that you have a total of 46 chromosomes. An egg has 23 chromosomes and sperm has 23 chromosomes. So you add those together and you're back to diploid. So this was a haploid cell, this was a haploid cell, back to the diploid number. All right, law of probability. This is when you multiply the chances. All right, so we could say, what are the chances that you are going to have 3 boys in a row? And so the chance of having the boy the first time around is 1/2. And then the chance of having a boy another time is 1/2. And the chance of having a boy another time is 1/2. So 2 and 2 is 4 and 2 is 8. So the chances that you will have 3 boys in a row is 1/8. All right, you could also use this law of probability when you're trying to figure out the chances of making a certain gamete. So let's say your genotype is this, and I want to know what are the odds that you're going to have a gamete that's little a, little b, little c. All right, the chances of having a little a out of big A, little a is 1/2. The chance of a big B is 1. And the chance of a little c is 1/2. So that means it's 1/4 or 25% chance. Okay, you can also use this. Let's say we're doing a cross, big A, little b, this times this. And I want to know what are the odds that I'm going to have an offspring that's big A, little a, big B, big B. And so I can look at just the A's. And so if I did this cross, whoops, I would figure out that 1/2 of the time you would get big A, little a. And then I could do the B one separately. And I would figure out 1/4 of the time you would get big B, big B. So 2 times 4, that tells me the chances of that is 1/8. So it's another way of using that law of probability instead of doing out a gigantic cross. All right, using Mendel's Punnett squares to predict traits of offspring. So basically, due to the law of segregation, like I've just been showing, you separate the alleles and you can write them on a line like this. So if my parents were big A, little a, crossed with little a, the odds of the gamete being a big A here and here, so I separate them. But for this parent, there's only the possibility of giving little a to the offspring, so I only actually need to make 2 boxes in my square. And then I want to make sure that you really use these terms correctly. Genotype versus phenotype. So a genotype are the letters, the alleles. So what are the odds that it's going to be big A, little a? 1/2. What are the odds that it's going to be little a, 1/2? And a phenotype is the actual trait. How is it distinguished? How is it seen? So let's say this leads to short fingers, and this one leads to long fingers. That's the expression of the trait. That's the phenotype. All right, patterns of inheritance. There's many patterns of inheritance. So I just wrote out a bunch of them to show. So law of dominance. If you cross one trait, so let's say that this is dark fur with a light fur, and all offspring come out dark. That's an example of the law of dominance, meaning that if a certain trait is present, it will always be expressed. It will always show up. So if all of the offspring here are dark, then the dark trait was dominant. All right, law of segregation I've actually already shown, but this is just that alleles separate in meiosis. And so that's the basis for, like, if this was my cross, my proof that law of segregation exists is that if I cross this with this, so let's say this is a brown and a brown, I actually could get 1/4 chance that they would come out white. So it's proof of the law of segregation that some of the offspring end up little b, little b. All right, incomplete dominance is when you get a third phenotype for heterozygous. So big B, little b. Let's say I use the one from above, brown and white, and actually I can't use that one. If big B is brown, and little b is white, let's say I get a whole nother color. Let's say I get tan. That's an example of incomplete dominant. It's not completely dominant when it is with the recessive. So if the heterozygous gives you a different trait, that's incomplete dominance. Codominance means that they are both dominant and they both get expressed. Here, I'm going to use that example with the flowers. So I could cross red flowers, and it's big R, with white flowers, which are actually big W, and you know that it's codominant because you do not write uppercase and lowercase, you give them both capitals. So the offspring would all come out big R, big W, and they would be red and white flowers, both colors expressed per petal. All right, monohybrid cross. So a hybrid means the two letters are different, and mono means it's just for one trait. So a monohybrid cross would be this, big A little a crossed with big A little a, and you can set up your square. And you know that you're going to get 25% big A, 50% big A little a, and 25% little a. So these are my genotype frequencies. And if this was a case of dominance, my phenotype frequencies would be that the dominant trait would be 75% expressed, and the recessive trait would be 25% expressed. All right, a back cross or a test cross. All right, let's say I have a brown fur and I don't know what the second letter is. It could be big B, it could be big B, little b. So what you do to test it or back cross it is you cross it with a homozygous recessive. And when you do that, if any of the offspring come out little b, the recessive trait, then that is proof that your genotype was big B, little b. And if you do the cross over and over and over again and none ever come out little b, you are pretty confident that it was homozygous dominant, big B. All right, linked genes are two genes on the same chromosome. So in humans, of course, we have linked genes. We have things that are on the same chromosome. You can have hundreds to 1,000 genes on a single chromosome. But we really do a lot of studies with fruit flies. And the reason why it's so much easier to study linked genes with fruit flies, first of all, they have a really short incubation time, like they have offspring every 14 days. So I shouldn't say that. They can have their offspring within 14 days. Within 21 days, they go through their entire life cycle. So you can see many generations in a short time. But they also only have 4 chromosomes. If there's only 4 chromosomes, chances are many of their traits are going to be found on the same chromosome, so they're linked. All right, a dihybrid cross. All right, di means I'm going to look at 2 traits. Hybrid means that both of them are going to be the heterozygous. So here's my cross. And when I do out the squares and I come up with the possible gametes, there they are, the 4 across this way. And then I would have the 4 this way. And you can go through, you can do all of your boxes. Again, I always recommend coming up with a list and making little marks like this to write in where you see those genotypes. And you're going to come out with a 9:3 to 3:1 ratio. And what that means is that 9 out of 16 are going to have both dominant traits. And 3 out of 16 are going to have 1 dominant, 1 recessive. The other 3 will have the other dominant and the other recessive. And only 1 out of 16 is going to have both recessive traits. All right, multiple alleles. This is when you have more than 2. Blood type is a great example to use here because you can relate to it. So the alleles for blood type are I, big A, I, big B. The I stands for immunoglobulin, which is a blood typing. So A means you make the A antigen and B means you make the B antigen. And then there's also a little I. And the only way that you can get O type blood is to be homozygous recessive. If you have either one of the dominants, like this, you would have A blood. And if you have both of them, you express both. They are codominant, so you'd have AB blood. All right, sex-linked. This is when a gene is on a sex chromosome. So usually we look at X-linked traits, because if you have a Y-linked trait, it's only going to be for the male. But X-linked traits, two of the most common ones that you see are hemophilia and colorblindness. And I would just put a big star here, because I think something that comes up really frequently is that they give you a pedigree and they ask you to go through it and figure out if it's sex-linked. So if a trait is sex-linked and a daughter has it, dad must. Oops. All right, and let me just explain that with a little pedigree here. So here's my daughter, and she has this trait. So X little a, X little a. And I go up to her parents, dad and mom. She got that X little a from her parents. So dad's other letter is a Y, which means that if he gave her that trait, he has to express the trait because he only has one X chromosome. Also explains why males are more likely to have sex-linked traits. They only have one X, so whatever they have is expressed. Whereas this female, her other letter could be X big A, and mom could not have the trait, but still pass it on to her children. All right, and here's the pedigree, and this one actually is going through hemophilia. So that is a sex-linked trait. So my advice is always to go around and do your X's and your Y's first. Now the circles are the females, so you give them all XX. The squares are all males, so you give them all XY. All right, once you've done that, you can go through, and anybody that has the trait has the lowercase. So there's a dad with it. Here is a son or grandson even. This is a girl that has it. So she's both X little a, and another one. Okay, so you go through and you do all of your letters of the ones that have it. Then I think it's easiest to go to the rest of the males. So this male doesn't have it, so he's X big A, and whoops, this one is also X big A. And then you can go around and figure out the girls. So this mom, number 2, had a daughter with the trait. So that shows she has to have a little a. But she has to have a big A because she's not filled in, meaning she doesn't have the trait. These 2 daughters, same thing for each of them. They have a mom with that trait, so they had to have inherited a little a from her, but they don't have the trait, so they get a big A. Same with this one. Dad could only give that X little a. And 9 actually has 2 possibilities. It could be X big A, or X big A, X little a. There's actually 2 choices for number 9. All right, when looking at data of offspring, how can you tell that 2 traits were linked? Using this data, so the reason why is your results are really off. Like drastically off. You're going to see it here in these numbers. Using this data, find the crossing over frequency for the following sets of data for the crosses. What does this information tell you? So see how the red hair with the freckles, also we've talked about red hair and freckles multiple times, so we already knew that this trait was linked. But red hair and freckles, you get 1,300. And then red hair with no freckles or brown hair with freckles, these 2 are really, really rare. So these 2 are due to the recombination, which means they're due to crossing over. And so if I want to figure out the crossover frequency, I'm going to put my recombinants over everybody, because the chances of something is always part over whole, and then I'm going to go times 100 to get a frequency or a percent. So the first thing I do, 202 and 180 is 382 divided by, let me just grab a calculator here. So 1300 plus 382 plus 1280 is 2962, and then times 100. So I do the math. And I get that they are 13. The crossover frequency is 13%. This also tells me a really important piece of information. It tells me that they are 13, it's called linkage map units apart on the same chromosome. So let's think about that for a minute. Here's my chromosome. Here's the one trait, the red hair, and here's the freckles. And they're 13 apart because the odds of them separating, the odds of crossing over was only 13%. If I had another trait and that trait had a frequency of only 5%, it would have to be closer together than that because the odds of separating them is so low. So if the crossover frequency is really rare, so let me just write this down, crossover frequency is rare, then the genes are very close. And if the crossing over frequency is much higher, then the genes are further apart. All right, moving on. Why do predictions not always lead to actual results? So sometimes you just don't get the odds that you were looking for. And so the first reason is that it is chance, and those numbers are just the predictions. It doesn't always work out. I'm sure you can all relate and know a family that has had 6 boys in a row. So the odds just don't always work out the way that they expect. But the other big factor is the environment. So, you know, that whole idea of nature versus nurture. So the environment in which the genes are expressed sometimes alters the expression of the gene. All right, DNA found in chloroplasts and mitochondria lead to specific proteins. In animals, mitochondria are passed on from the egg and not the sperm. And in plants, mitochondria and chloroplasts are passed on from the ovule and not the pollen. How are these traits not inherited in typical Mendelian genetics? So these are not passed on by dad. So no Punnett squares. You don't need a Punnett squares. It goes straight from mom to the offspring. So you simply look back and like the traits of mom are going to be expressed in the traits of the offspring, and the traits from the dad really aren't shown because they aren't passed on from dad to mom. All right, phenotypic plasticity is a term to describe the effect of an environment can have on the expression of genes. Individuals with the same genotype may express different phenotypes based on the environment. In class, we learned about epigenetics and the reasons why genes may be expressed differently in individuals with the same genes. Explain. Okay, so there's actually multiple things that we want to talk about here. First of all, we want to talk about, well, this is a new word that I just found the AP including, so I just want to make sure that we use that word. So that word means the differences in how organisms that have the exact same DNA could express the traits differently. So we looked at things like their diet and the amount of exercise that people were getting and the stress and chemicals that they were exposed to. All of those things could end up having an effect either to inhibit or to activate a certain gene. And we call the different expressions of genes epigenetics. And something that was really important that we talked about in epigenetics was methylation. So I want to just go back to that. When you methylate something, you're adding methyl to it, and methyl is just CH3, which is very nonpolar, which means it's going to hide. It's going to be hydrophobic. And so if you put a methyl tag on something, you are essentially turning it off. You are hiding the gene. Now there's two things that you can methylate. You can methylate a gene, which is like an on-off switch. And so if you methylate a gene, it is now off or it is now silenced. But you can also methylate the histones. Histones are the proteins that DNA coil around. And so if they are methylated, then they will coil very tightly. And if they're tight, they are less likely to be expressed. And DNA that is coiled very tightly is called heterochromatin. So heterochromatin is not likely to be expressed, which means you're not going to be able to have transcription factors get in there and make the RNA. But if it is very loose, then it is more likely to be expressed. And we call that euchromatin. All right. There are multiple errors that can lead to genetic conditions. Explain. All right. So the first thing that I think most people think of are mutations. So we learned about 3 different types. We learned about silent, which is going to give you no change in the amino acid. So it gives you no change and no difference in a genetic condition. But if you have, if you have a missense, then you end up coding for a different amino acid. If you code for a different amino acid, you can change the overall shape and the overall folding that occurs. And if you change the overall folding, this is a great example of what happens with sickle cell anemia. So a single change in a nucleotide could actually end up giving you different mRNA, which codes for a different protein, which gives you a different shape, which makes your cells not work. And then the last one was called nonsense, and nonsense coded for a stop. So that's definitely going to alter your overall protein that you make, and it might not be functional at all. All right, another thing that can lead to genetic conditions are, so this was a mutation in the DNA. You can also have mutations or changes in the chromosomes. So a chromosome could be missing, or you could have an extra chromosome, or you could have a broken chromosome. All of those things are also larger mistakes that could lead to a lot of drastic genetic conditions. All right, and so the last one that I have here is Down syndrome, because I think it is a great example of a genetic condition that is due to an altering of the chromosomes. And so what happens is non-disjunction. So non-disjunction is the failure of sister chromatids to separate in meiosis. All right, so in meiosis 2, the sister chromatids that have already been separated from their pairs are now supposed to go to each side. And so you would have one over here and one over here, and your gametes would have one of each. But instead, what happens with non-disjunction is when this goes to separate, whoops, arrow's going the wrong way. When this goes to separate, 2 end up over here and none over here. So nothing and 2. And so if this cell, which has, I'm just going to put +1, when this cell ends up in the egg, So here's my egg, it's N plus 1. And here's my sperm, which is N. I end up with a zygote. That is 2N plus 1. So when you have an extra, if the extra is chromosome number 21, it's not triploid because I don't want 3 of every chromosome. Trisomy. Trisomy 21, 3 of chromosome 21. Then that leads to Down syndrome. All right, let me just go back here and make sure I didn't miss anything. All right, so look back through everything that you have. I know this was a much shorter unit. Take the time, make sure that you do AP Classroom and see what questions that you want to go over. Bye.